Okay. So welcome everyone to the lecture. Thank you, organizers, for inviting me. This is my second visit to IIT Park. I think 6 years ago I also came here. I gave a series of lectures that was an old building, which yes, we have a brand new building, and I'm very happy to come here. And thank you for visiting my lecture. I was very afraid that nobody would come, nobody would sign, but the audience is big, so thank you so much. Okay, so we will talk about stochasticity and randomness in chemistry, chemical biological processes. That will be today's lecture, will be introduction. And I want to emphasize that this is a brand new class that I didn't teach yet. And another important issue is, as you already heard, this is extremely multidisciplinary, right? I'm professor of chemistry, my friend is collaborator of mathematics, this is mathematics. We'll talk about biology. So for this reason, the audience, as far as I understand, also very multidisciplinary. So for this reason, I will try to be as simple as possible. Despite this is the department of mathematics, I will try to have the minimum mathematics. So that the understanding is more important. But feel free to interrupt me anytime, to ask questions or something. So as you probably, one of the only few good things from the COVID time, I understand that education, the value of education of person to person. Okay? Zoom or whatever cannot substitute it. Education is two-way street. It means lecturer presents, the students ask, and lecturer adjusts. Okay, so please do, let us do it a good education. Okay, so this is introduction. So first I start with my personal introduction. I was born in Ukraine. That was, in those times it was still one country, Soviet Union. This is my family picture. This is me, so 3 years old. In those times, color photos didn't exist or was extremely expensive. I don't remember exactly. So I graduated with a Master of Science in Chemistry from Moscow University in Russia. This is Moscow University in Russia. It will be dark, will be better. Yes, like this is okay, yeah? So this is what the best university in Russia, one of the best in Europe. So I got my PhD in chemistry from Cornell University in Ithaca, New York. Like, if you've not been there, strongly recommend you. It's a very beautiful place in the mountains of New York, 5 hours drive from New York City. One of the most beautiful campus, university campus I have seen in my life is that Cornell. Strongly recommend it. Not only good education, also very beautiful. Cold, a lot of snow, but it's very beautiful. So I'm professor of chemistry, physics and astronomy by one department called physics and astronomy, and all other department is chemical biological engineering at Rice University. And so this is my personal dedication and introduction. And I will try to figure out who you are, maybe we'll learn the name. Okay, so this is our main goal. Okay, before doing something, you need to think what you want to do, right? You don't jump into the river. You should know how the temperature of the river, whether it doesn't have crocodiles or not. So we need to think again. So this is our main goal. We'd like to understand the role of stochasticity in chemical biological processes, but we're not interested in understanding these things per se, because it's interesting. It's interesting, it's cool, stochasticity, randomness. We actually have a more, I would say, fundamental goal. We want to understand biological and chemical processes mechanism using this tool. Okay, so it's the final ultimate goal is actually to understand the mechanism of this phenomena through understanding stochasticity and randomness. That's what the goal. Okay. So the idea is that we will try to develop a theoretical framework a little bit, but we will actually do something what in America called learning by example. We'll consider several systems. We'll discuss the systems and assuming, hoping that looking at the different systems from a pattern, we will recognize the role of stochasticity. This is the idea. And something that I already mentioned. So I will do a lot of hand waving. Okay, we'll try to be as simple as possible. We'll try to have a minimum mathematics, despite the fact that this is the part of mathematics and the topic has a lot of mathematics. The point here is to understand. The point is not to overwhelm you, to show that I know some mathematics. No, the point is for you to understand things. And this is a lesson for you. Whenever you present something, do not show mathematical calculus as minimal as possible. Okay? The idea is more important than the way it's done. Okay, so the point here is we'll try to do intuitive understanding so that we can understand the role of stochasticity or randomness. So we will start with chemistry. A little bit, of course, chemistry is a huge topic. Chemical reactions. Probably all of you had chemistry in high school, right? Right, and probably many of you had chemistry as a student, right? And not many of you. So if you remember, chemistry is very interesting topic, very fascinating topic, right? And something that we remember from when we were children, that this magic, right? You mix up two transparent compounds, liquids, and then there's a green color come out. Or you're mixing them up and then there's a foam come out. So that's what probably they show you frequently to attract people to chemistry. So chemistry is a fascinating topic. But from the point of view of a little bit more scientific approach, this is a transformation of matter. The transformation of matter when one type of molecules is changing into another type of molecules, and typically you break the bond or make the bond. That's what's happening during the chemical reaction. There are four main types of chemical reactions. This is approximately. So one chemical reaction is the synthesis from two blocks of atoms. You're creating a bigger block. Another type is decomposition from a bigger block. You're creating smaller segments. And then there is this is called displacement or substitution, when you're substituting blue one with the red one, as shown over here, or double displacement, double substitution. This is generally many. There are many combinations. Not important for us. What is important, more important for us, is that if you think at the molecular level how chemical reaction is happening, they're happening due to collisions. Molecules collide, and then something happens. But it's important to understand there is a huge number of collisions. Typically like billions of collisions, only one of those is successful. There are many collisions, but only few of them successful, have enough energy, and so on. So my analogy here is relatively unpleasant. You have to pay something that it's not, the cars can drive after this, okay? Nothing serious happens. That's most of the collisions like this. And occasionally you have something like, I have no idea, but the both speeches are from America. I have no idea how it ended up, but it probably ended up better. So this is like chemical reaction. This is chemical reaction happening. It's rare, but you have to have enough energy. Cars have to drive with enough speed. Drivers has to be stupid enough, and so on. So it's something like this. Good. So collisions, molecular collisions. As I already mentioned, not all collisions lead to chemical reactions. So only those that molecules have enough energy, speed of the cars, proper geometry, and that's what reaction happens. So I emphasize the word random. Okay, but collisions are random. Okay, collisions are random, and it's also random to have enough energy or proper geometry. It's a random thing. So which means this randomness or stochasticity, we will use these two words as synonymous. Randomness or stochasticity is the most critical aspect of all chemical reactions. So chemical reactions, what we have to remember, are random, very random. So you might ask me a question. So what is the time before chemical reaction will occur? And it's not so easy to answer, because essentially the answer is it depends. Okay. So what is plotted over there? This is from experimental data. This is the concentration of hydrogen peroxide. So hydrogen peroxide can associate with water and oxygen, and this is the function of time. Do you use hydrogen peroxide in India, in pharmacies? In America, you can go and buy it. It's usually in black bottles. It's antiseptic. You pour in something and you can see bubbles come because of oxygen. I don't know if you use it. Probably use it. It's very useful antiseptic. So this is the reaction. Hydrogen peroxide dissociates into water and oxygen. So this experiment, they're able to look at the concentration of hydrogen peroxide as a function of time. The function of time is decreased because of reaction happening. But this plot also simultaneously means that this is, you can also think about it as the time before reaction happens. So this plot tells you time before reaction happens. Imagine that you look at a single molecule H2O2 and you wait until reaction happens and you repeat the experiment again, many times. And you will have a plot like this. There will be very different times of reaction, many different types of reaction. So, but you heard chemists telling you this is the rate of reaction. You took some chemical classes because the rate of reaction is inverse of mean time. In this process, there will be some mean time. If you repeat it many times, there will be some average time. And that's inverse of this. Mean time is called reaction time. Okay? But the important to understand that it's not a one time for reaction. Chemical reaction is a random process. There are many different types. There's a distribution of reaction. Okay, so the fact that these reaction times are random, you can actually better see from so-called single molecule experiments. Yes, please.
In the previous picture, is that when the experiment is conducted once you're measuring the concentration?
No, in this case, it was done in a typical high school. I don't know. You took a beaker, and suppose you have spectrophotometer which can measure the concentration of H2O2 from the density, and you will see how the density changes, something like this, for example. And here I will show you single molecule picture. So single molecule picture is that frequently molecules that react or products that produce this can fluoresce. Okay, and this experiment that I show you here, the reactant, the product is fluorescent, but reactants are not. So in other words, when reaction happens, you see this spike, right, due to fluorescence. So this is real single molecule measurements, and you can see this is the time between reactions. This is the time between reactions. And you see it's not the same, it's really different. And some of them happening faster, some of them slower. And that's a single molecule level. Okay? So you can see that there is always distribution of reaction time. And when chemists say reaction rate, it's inverse of the mean time. So all distribution, chemistry is random. That's what we have to remember. Chemistry is random. Good. So let's talk a little bit more. Okay, by the way, ask me any questions, interrupt me. I have a tendency to speak fast. I will try to be slow. Do you understand me? Do you understand my accent? Because I'm not American, I speak with Russian accent. You understand, right? Good. Yeah. So when you do like this, it always confuses me. In America, I always do. So I had at first my Indian student, I was explaining him something. He was doing this, and I thought he doesn't understand, so I explained him 3 times. Before I understood that it's me, stupid. Anyway. So biological world. Biological world is a mean, right? So it's a lot of very different strange phenomena. It's a remarkably complex and broad spectrum of phenomena. And we understand that this is evolution shaped all these things here to produce this remarkably complex spectrum of phenomena. But I'd like to concentrate on a few aspects of this biological world phenomena, and 3 of them. One is I can claim, and you probably agree with me, that one of the most striking properties of biological system is the ability to sustain highly efficient, remarkably precise, consistently robust function. Okay? So let me go and to show some examples. So biological process, as you understand, there is no process can be 100% efficient, right? There's no way. So biological process is quite efficient, and people argue that the typical efficiency of biological processes is ranging between 10 to 50%. And you understand that the process cannot be 100% because you need to do something else to keep it. So for example, we need to do something to have our temperature to be warm, okay? So this is a waste of energy. So I don't spend all my energy walking or doing some, because some energy is used to keep me warm, right, and something else. So, but I'm still arguing that they're highly efficient. Why? That's because humans eventually make devices, in many cases, which mimic biological systems, but they're not as efficient as those produced by biological systems. Yeah. Okay, good. So one of the sources of energy in biology, so it's very complicated topic, and again, I'm trying to not to go into detail. So one of the most important molecules, which is called energy currency, is ATP or adenosine triphosphate. So cells produce these molecules, and then in different places, different locations, hydrolysis of this molecule produce energy. That's how biological systems use energy. This is a very simplified scheme shown over here. So ATP fuels essentially most biological processes. We can actually do some very simple estimates. So estimate what the efficiency of biological processes could be. So the way it works essentially, some of the biochemical cycles that we have as well as photosynthesis, they actually produce molecule of water. The synthesis of molecule of water produces a lot of heat. But then cells use this heat or energy to produce ATP. So what people know that if you're synthesizing 1 mole of water, that amount of energy can be produced. Water is very strong bonded molecule, so it produces a lot of energy. And then people saying that there's approximately 3 moles or 3 molecules of ATP produced. Each of them has this typical heat of reaction and typical physiological conditions. So what you do, you 3 times this will be 21.9. This is energy. Essentially dividing this by this will give you the efficiency. So let me repeat again. So the cells or living system synthesize water, 1 molecule of water, and that energy is produced. But for cell functioning, only 3 molecules of ATP simultaneously produced, and that's the way to estimate efficiency. That's not the only way to estimate efficiency, but it gives you approximate idea what is the efficiency of biological processes. Now, there is an important word homeostasis. Have you heard word homeostasis? Are you taking biology? No? Oh, surprising. Okay. So homeostasis, it's simple. It's relatively simple. This is a tendency of living systems to have almost constant physiological parameters. For example, blood pressure, sugar level, body temperature. This is foundation of modern medicine, right? We know when we are okay, 36.6 degrees of Celsius, right? If it's not, we are wrong. I mean, we're sick. Blood pressure, 120/80. Sugar level. This is the foundation of modern medicine. That's the way the doctors work, okay? They're not smart enough. They only look that when you go to do tests, any deviation from those fixed values is a sign of something. So this phenomenon called homeostasis, okay? And you might ask me the question, why organisms have homeostasis? It's not clear, but most probably this leads to the most efficient functioning of organisms, most probably. Okay, let's lead to the most efficient functioning of the systems here. So I'll show you one more picture. So obviously this is the American way of measuring temperature, right? Because degrees and Fahrenheit, right? So in Fahrenheit, 100 Fahrenheit is considered to be approximately normal temperature. That's how this scale was introduced. Although Fahrenheit was wrong. He measured temperature of his wife. Anyway, so in 100 over 20, and this is for example blood sugars, right? Typically 100 milligrams per liter is borderline. You go and take some tests and so on. If it's more than, there's no problems here. Some other tests here. Okay. The point I'm making here is that biological system did something to keep it almost constant. That's the point. Okay. Not that it's interesting to have this constant temperature. They keep blood pressure constant in healthy organ, also sugar level healthy and so on. So biological system doing some extra things to do things almost precise. Okay, that's my point. Robustness. Robustness is the ability of plants or organisms to survive and to flourish in different environments. Okay, I have an example which probably will be closer to people here. So have you been in Chandigarh Rose Garden? Some local people did. It's very good. It has some roses. Don't recommend you, right? The same, exactly the same roses you can find in Moscow, Central Park of Moscow. The same roses, I checked the same type. Johannesburg, South Africa, I'm pretty sure if I find the pictures in Houston. The same plant in different places with different climate that shows the robustness, right? Of course, there is a limit of robustness, right? You can probably find monkeys here, but not in Russia. It's too cold for them, right? But still, biological systems show remarkable robustness, okay? Babies born successfully here, Africa or United States, that's a robustness, okay? So, it's a remarkable robustness. And let me show you this robustness on more microscopic level. So, as you probably know, proteins are the main building blocks of biological systems. And for many proteins to properly work, they have to fold into correct structure. And but frequently this folding is not correct, so-called misfolding. So apparently nature come out with a solution because it created another molecule called molecular chaperones, which also protein, which help the original proteins to properly fold. This picture shows this thing here. So this is your correct so-called native state, the one that you really want, the one that will properly work. But there is you see, there is some traps here. So the chaperones help you to untrap and to go there. This reminds me, so like 3 days ago, I was in Dehradun and I took to Uttarakhand state, and then the driver took me to a Mussoorie place, right? The road is very narrow, so the point is two cars almost cannot in some places go through, right? So what they do, so the one car back up, right? To widen things. It's exactly a more molecular, more smarter way. Anyway, so yeah, so this is another sign of robustness in biological world. This molecular chaperone later on formation of this intermediate, these folded things here, and this allows biological functioning to proceed properly as it should. Okay, so another interesting phenomenon in biology is existence of thresholds. So many biological processes happening after some key components or key players reach some specific number, threshold, and then it's happening. So cell division, one of the most complicated and important processes is cell division. And if you look at cell division, there are several so-called checkpoints. So this is a cycle of, have you seen these cell division cycles? So people who do biology probably saw it. So essentially, there are several cell checkpoints. Suppose we are in this first state. So there is a checkpoint which checks if the cell is big enough. If you build enough proteins that you need it for separation, then yes, go ahead and you go to the next stage. If not, you continue until you do it. Then there is another checkpoint over there. Here's DNA synthesis is taking place. DNA is a place where all the genetic memory here. So they're checking that everything is done correctly, no mistakes, because if there is mistakes, your cell will get mutation. And if it's okay, then there's go ahead and you move to the next stage. And then there's the mitosis checkpoint where cell division happens itself. It checks that the chromosomes separated properly, that everything is in order, and so on and so on. Again, there is existence of threshold. There's the threshold. They check something, some number of some proteins, signal proteins, which tell you that you can go ahead. There's another example. So in every cell that we have, there is a protein produced, P53. P53 is extremely important protein. Mutation in this protein is responsible for 70% of cancer. So this is a protein which is known as a genetic guardian, stress guardian. So if cell feels some stress, it starts to produce this protein, there's stress, more stress, and then suddenly when the number of P53 reaches some number, threshold number, boom, cell dies, goes through apoptosis. Okay. The question, so it's quite precise phenomena. Okay, cell dies only when it reaches some number.
So if it crosses the threshold value, then
That's it.
okay. even if cell didn't die then, it's going to recreate, replicate of other cells.
No, this cell will die, some other will survive. But you could, my point is, but you could imagine another scenario, right? That there will be some probability to die from any possible amount. But it doesn't. It only happens when you reach some level. Okay? Something like this. Which is another indication of preciseness in biological system. Very precise. Okay, so another interesting examples of precision in biology. I wanted to find something unusual. So this is a beautiful plant. You know, any of you travel to South America, you know? So if you have a chance, you go to South America, there is a desert called Atacama Desert, Peru in Chile. And it has a plant. This plant is called Azorella compacta. So this looks like this, and it's growing gradually. So what is interesting is that this plant grows exactly 1.5 centimeters per year, exactly, with a precision up to millimeter. 1.5 centimeters. 1.5 centimeters. Of course, you can argue, okay, this desert climate is more or less constant all the way around, so maybe still it's amazing precision. It's amazing precision. So it's amazing precision, and by the way, this plant can also flower. And scientists, when they look at this plant, they found some of those plants are 3,000 years old. And the way they figure out it, they measure the thickness. Okay, so I keep saying that precision of biological process is surprising, right? I give you a couple of examples. Surprising. Why I'm saying this? That's because to a large degree, biology is many chemical reactions. Biology, to some sense, it's huge number of chemical reactions. But we just talked with you a few minutes ago that chemical reactions are random, right? So the question comes, how many random events lead to something more or less organized? So traffic in India maybe not a good example, but still not so many accidents, right? Extremely random from my point of view, but somehow they're able to, okay, maybe it's not a good example. But let me give you a simpler example. So imagine you have one reaction, let's call it chemical reaction A. And let's say this is the reaction time, distribution of reaction time for this reaction. Let's have separately another reaction, chemical reaction B, and the distribution of reaction time. Let's have a phenomena assumed that which will have both reaction A and reaction B. This is what we would expect, a broader distribution, right? One is random, another random. If you combine, it will be more random. It's naive expectation. So then now imagine thousands of those reactions, right? You would think that biological phenomena would have a very broad distribution, very noisy, very random, but it's not. I've already showed you with your example, right? So that's what we wanted to understand how it's happening. Okay, this probably should be actually the first slide. We talk about stochastic process. We should define what is stochastic processes. Okay, the stochastic process is not every process which on-off is stochastic. So the stochastic process is a phenomenon in which some quantity varies in time in a very complicated and irregular way. That's an important statement here. If there is some phenomena which varies, but you can see the pattern, that's not a stochastic process. You understand? That's an important thing which people frequently forget. Stochastic process is something that you cannot understand the pattern of this variation, so you have to do a lot of time averaging to get it. So in some sense, you can see this process can be viewed as an ensemble of processes. So let me give you two examples. That's not a stochastic process. It's day, night, it's not. We typically know when day will come at night, right? Getting salary, hopefully, is also not a stochastic process. You get it regularly, okay? So just want you to understand the difference in stochastic process versus those processes which change, but we know what is, change of the government in India, it will change, right? More or less random, but still, it will change within democratic government. Okay, so we have some preliminary conclusions with you. I argued, and hopefully you agree with me, that this randomness of stochasticity is important for chemistry. systems which are in some sense mini chemical reactions, despite naive expectations, is also is quite precise. So somehow biological system in many cases can tune this randomness. I don't know the proper word. Stochasticity is important for biological processes, and it's interesting to understand how it's happening. And again, I argued something that at the beginning we would like to understand stochasticity and randomness, but I want to emphasize not because of its interesting topic, but because it might give us a backdoor entrance to understanding the world, microscopic world of chemistry and biology. Okay, this is the reason we're trying to do. Yeah, so this is the plan. So we'll be total 12 lectures. So today the first one will be second lecture today. We will do it this way. We will try to discuss some chemical reactions, a little bit more quantitatively, chemical kinetics. We'll discuss a little bit catalysis, some geological chemical rates law. We will understand some other tools, some theoretical tools needed to understand it. But the biggest part of this lecture will be learning by examples. We will look at specific systems. Okay, hopefully finding some pattern. Hopefully this is not stochastic, at least to find that's the idea. Okay. Okay. So yeah. So this is essentially, for the first time, I don't want to overwhelm you, and I was trying to be slow. Feel free to ask me any questions. Wasn't too much biology or not? I was trying to chemistry. Again, I want to emphasize the audience is multidisciplinary, the topic is multidisciplinary, so I'm trying to be as simple as possible, which means that some of the things that I put under the rock, right, are just in black, but it's here. Any questions?
So, stochastic behavior, for example, in any kind of biofilaments, it gives the collective behavior as well. Like,
yes.
Is it like, for example, the catastrophe in filaments?
Yes.
Is it related with
right. All right. All of this is chemical reactions, so it's stochastic phenomena, and there will be some collective behavior. Sometimes we don't understand what it is, but it is a result of this thing. So it's not a regular phenomenon. It's a random one.
So what gives us an idea that stochasticity will be the convenient tool to understand the microscopic best?
I was trying to argue because biology in naive way is chemistry, mean huge amount of chemistry. And chemistry is stochastic phenomena. That's the idea. It's not guaranteed that will fully clarify the phenomena, but it seems to be, given the fact that stochasticity is one of the most critical aspects of chemical reaction, chemical processes, that should help to understand biological phenomena. That's the idea. Mm-hmm.
Sir, I have one question. From some slide with MFPT and drawn some noisy paths. Like some noisy paths are there, the critical threshold level. In the MFPT part. This one.
On the right?
Does this distribution, make question that is this distribution depends upon the type of the noise or how much noisy the system is? Is this MFPT distribution actually?
Yes, understand. Yeah, so it depends on the process typically, might depend on phenomena. In many cases, people try to model it as a Gaussian noise, but it's not real. So the way people typically do, they assume some model, the model predicts some distribution, and they see if this will fit data. But data could be more complicated. The distribution is a reflection of underlying mechanism which we don't fully understand yet, but that's something like this, okay?
So it may have dependency on the.
It will definitely have a dependency. It will.
So here it is like Gaussian technology.
It is just for illustration purposes, but it could be, most probably Gaussian is not the right answer.
Yes.
Most probably it's a combination of exponents, something like this. This is not real. It depends on the mechanism. It depends on the mechanism of this process of this threshold. Because, as I said, it's threshold, but strictly speaking, there's a little bit variability in thresholds and so on. The phenomenon is typically more complicated.
One quick question. So you mentioned like stochasticity, through stochasticity you can understand chemical processes, which leads to understanding of biological processes.
Yes.
Is there any counter example for this, like where stochasticity fails to?
I don't know any examples like this. I don't know, but that's still not a guarantee that you will. As you probably know in mathematics, right? We frequently have formal solutions of something, right? But they're completely useless for practical application, right? So it could be this story. Formally correct, but so. Okay.
Fine, then we end up this section. Thank you, sir.
If some of you wants to just come and ask me, some of you are afraid to ask in audience, that's fine. One-on-one, that's fine. Okay, thank you.
Like we have still some more minutes. We decided this lecture for 1 hour. So what we can do, we can formally introduce yourself to the speaker. Yeah. Where you from, what you are working on. That would be a good idea. Okay. So let's start with Nisha then.
Yeah, that's a good idea. That's a good idea. Would be nice. I will try to remember your names.